Welcome to the Chapter 12, Section 4 podcast on mutations. And what is a mutation? They are changes in genetic material such as RNA and DNA. And mutations can produce two types of changes in chromosomes. First one being mutations that produce changes in a single gene are known as gene mutations, and we'll get to those. And mutations that produce changes in whole chromosomes are known as chromosomal mutations, and we'll talk about those two later on. So let's talk about gene mutations first. The gene mutations involve a change in one or a few nucleotides, and these can be known as point mutations because it occurs as a single point in the DNA sequence. And we have several types, which we'll get to in detail. So substitutions with a substitution usually affects one single amino acid. If you look right here, we have a cytosine right here, and a thymine gets substituted instead. And if you notice, what it does is it changes this amino acid right here, which could adversely change the structure of the protein. If you notice, this amino acid and these two amino acids are not changed. They're the same down here. Now, if you have an insertion or deletion, it can be a lot more dramatic because it can cause a shift in the grouping of the codons. And remember, codons are groups of three bases together that code for one amino acid. We call these changes frame shift mutations because it shifts the whole set of codons. Let's do an insertion here. Well, actually, let me make this point first, is that when you have a insertion or deletion, a frame shift mutation, that the change is in every amino acid that follows the point of the mutation. So from this point on, all these amino acids here are going to be changed. And sometimes it could alter protein so much that it's unable to perform its normal functions. Often this could lead to adverse effects that often can be fatal. So let's look at that insertion again. Here we have a cytosine, and instead what we're going to do is insert a thymine here. And you can see that changes this this first amino acid here and pushes this C to right here. So you can see all these amino acids are changed. So if you look, this is what we should have, but this is what we get. And here's that T on the end. This T is that T right there. So everything's been shifted. Now in a deletion, rather than inserting something, what you're going to do is you're going to lose a single base, and that's also going to shift the frame. Now this isn't DNA, this is actually a sentence, but watch what happens when we delete this H. This reads, "The fat cat ate the rat." Now if we delete the H, this is what we're left with, and that's you can't even read that. And you can notice here's the last two nucleotides right there. You can see, bases, nitrogenous bases. You can see them right there, left at the end. Now, chromosomal mutations involve the number or structure of chromosomes, and it can include 4 different types of mutations. Here's a deletion, and a deletion involves loss of all or part of the chromosome. And you can see right here, here's B. This is part of the chromosome, and here it's lost, and that genetic information is lost there, and this could vastly alter the organism. Here's a duplication right here. Hold on. Duplication produces extra parts of the chromosome. You can see B is duplicated, so there's 2 segments of B. Cows, which are ruminants, meaning they can eat and digest grass, they were created as a result of duplication of a gene in a chromosome. An inversion is when you reverse the direction or parts of a chromosome. And if you notice right here, we have B, C, D, E, this section right here. And what's happened is it's been reversed. Here's B over here, so it's B, C, D, E going in the opposite direction. That's an inversion. And we could also have translocation, and that's when part of the chromosome breaks off and reattaches. So once again, if you look at D, E, and F here, it's left the original chromosome and ended up on a different chromosome over here. Now, most times, mutations have little or no effect on the gene expression. Often it occurs in an area where there's no gene to be expressed. However, sometimes it could lead to causes of genetic disorders such as sickle cell disease and cystic fibrosis, and these are both point mutations. One small substitution occurred which led to sickle cell disease, sickle cell anemia, and in that disease the red blood cells, when they are in a acidic environment, sickle and can no longer carry oxygen, which then causes the person to have shortness of breath, and it can be a very debilitating disease. But sometimes mutations can be beneficial, such as this beautiful looking leopard here that is a black leopard, and that's a result of a mutation that can lead to exotic new organisms, or organisms that are better able to survive, or maybe provide traits that are beneficial. Mutations have led to organisms that are immune to disease, and that can be very important when you have plant life. A beneficial mutation might allow an organism to become smarter or faster or stronger or bigger, and that's why mutations aren't always negative, quite often very positive. Now, somebody asked, what happens if in meiosis 2 the cells don't actually divide? And that leads to what's called polyploidy, which is where an organism has an extra set of chromosomes. Not an extra chromosome, but an extra set. And here's I think this mouse is from Argentina, and it's a certain one found there. It's native there, and you can see it has a mess of chromosomes because it's a tetraploid. It has 4 N chromosomes, which is double the chromosomes that its ancestor had. And often polyploidy plants actually are viable, and they continue. In fact, bananas and some citrus fruits arose because of polyploidy.